Hello everyone. In this lecture, we will talk about gluconeogenesis. So in our previous lectures, we have talked about the process of breaking down glucose to produce ATP. And now we will look at the opposite reaction, which is the synthesis of glucose in our body. According to laboratory standard, our blood sugar level should be around 4 to 6 millimole per liter during fasting. But when you have a blood sugar level lower than 2.5, then you have a condition. It is called hypoglycemia, which means the lack of glucose in your bloodstream. So for those who have diabetic patients at home, you will be trained to identify these symptoms of hypoglycemia. So this happens when the patients took the insulin but they forgot to take their meals. Or this can happen to you as well if you skip a meal or two. Initially, you will feel a bit tired, confused, and you seem to have difficulty recalling your memory. So all these are the symptoms of lacking sugars and energy. You might have as well having ataxia, which is a loss of body control. So you become more clumsy and even a personality change, such as becoming more irritable. So if you wait for a bit longer, more symptoms will start to develop. For example, you will have hemiparesis, which is the loss of feeling as your nervous system is impacted due to the lack of glucose and energy. You experience increased anxiety, palpitations of heart, and tachycardia, which means an increased heart rate, and many other symptoms as well. So eventually, you could go into coma and even death because of the lack of ATP. So it's very important for us to maintain a normal range of glucose in our body. But why is glucose so important? They are important for at least two reasons. Number one, the brain uses glucose as the predominant source of energy. In fact, our brain uses up to 3/4 of glucose metabolized every day, which is equal to about 120 grams per day. And for the red blood cells, glucose is the only molecule by which they can produce energy. And that's because red blood cells do not contain any organelle, such as nucleus and mitochondria, and as a consequence, it can only rely on the glycolysis of glucose to produce ATP. So showing here, you have the distribution of glucose in our body. You can see that there is around 20 grams of glucose in our body fluids, which includes our blood plasma and the interstitial fluid. But we have even more glucose stored in the form of glycogen, which is found in our liver and the muscle, and you have roughly about 190 grams. So with this amount, it can sustain us for roughly 1.5 days, which explains why we need to ingest food on a daily basis. So when does hypoglycemia happen? Well, it happens when we are under starvation and during or after exercise, when we have exhausted all of our glycogen supply. This is when you will experience muscle cramps. And these are the learning outcomes for this lecture. First of all, we'll look at what is gluconeogenesis and when does it occur, and then we'll describe the role of Cori cycle and the alanine cycle in gluconeogenesis. And then in the second part, we will identify three irreversible steps of glycolysis and explain how they are overcome in gluconeogenesis. And last but not least, we'll look at the control mechanism of gluconeogenesis. So what does gluconeogenesis mean? Well, genesis means creation of something. Neo means new, while gluco means glucose molecule. So it is the creation of new glucose molecule. Gluconeogenesis normally happens 2 hours postprandially or after meal. And this is to make sure that we have enough glucose to run all basic functions of life. And obviously, we can't synthesize glucose like plants do in photosynthesis, and we need to convert it from other molecules in our bodies. And the main place of this process happens in the liver because it is where we have the full range of enzymes to perform gluconeogenesis. So what are the sources or what are the molecules we can derive to make new glucose molecule? First of all, we can use pyruvate or the lactic acid, which is the products of glycolysis. We can use glycerol, which is the building blocks of triglyceride. We can use the intermediates coming from the Krebs cycle and even amino acids. So you have seen this diagram before. It is the major metabolic pathway in our body. And so far, we have learned about the metabolism of carbohydrates, which includes glycolysis, the Krebs cycle, and the energy production involving oxidative phosphorylation. So essentially, this pathway is going down, while for gluconeogenesis, we'll be moving up the pathway to produce new glucose molecule. However, it is not a direct reverse of glycolysis, and it is not as easy as you see, and you will see why in a moment. As I mentioned, liver is the major site of gluconeogenesis, and it also happens in the kidney, and to very little extent happens in the brain and muscle. So the whole purpose of gluconeogenesis is to maintain the sugar level in the blood so that our brain and our muscle can function optimally. Okay, you have learned that at the end of glycolysis, you get pyruvate. Pyruvate will be decarboxylated and enter into the Krebs cycle, which require oxygen, and you will get carbon dioxide, water, and ATP production. However, when there is a lack of oxygen, pyruvate is converted into lactic acid. So this happens when you have, let's say, a very heavy exercise. But as you breathe in more oxygen afterwards, now you have the oxygen for the Krebs cycle to resume back, producing more carbon dioxide and the high energy molecules. As a result, the concentration of pyruvate drops, and lactic acid is converted back into pyruvate by the enzyme lactate dehydrogenase. So now we can use that pyruvate to synthesize glucose molecules. But the problem is that our muscles do not have the enzymes needed to synthesize glucose, and the liver is the only place that we can synthesize glucose by using pyruvate molecule. So we need to transport pyruvate from the muscle into the liver for gluconeogenesis. And this process is known as the Cori cycle. So showing here, you have the Cori cycle, which refers to the conversion of lactic acid in the muscle into pyruvate in the liver, and from pyruvate into glucose molecule, followed by the metabolism of glucose back into pyruvate in the muscle. So from lactic acid into lactic acid, you have a cycle, which is the Cori cycle. And now you might think that the lactic acid can be transported through our bloodstream to the liver for gluconeogenesis. But we have a problem because lactic acid is acidic, and this will reduce the pH of our bloodstream. So can you still remember what is the pH of our bloodstream? Well, the answer is 7.40, and it is tightly regulated in the range of 7.35 up to 7.45. When it's lower than this value, it is called acidosis. When it's higher than 7.45, it's called alkalosis. And in both scenarios, it can kill us. So for example, when the pH is lower than 7 or higher than 7.8, all our enzymes around the body will start to denature or losing their activities. And it is not just about the enzymes, because all other proteins such as structural proteins, antibodies, channel proteins, they will be affected. So if lactic acid is accumulated in our bloodstream, it is really dangerous. So what do we do? Thankfully, our muscle do have the ability to convert lactic acid into pyruvate under an aerobic condition. So once you catch your breath after the exercise, your muscle will convert those lactic acid back into pyruvate. Well, you might think that this makes no difference because pyruvate is still an acid, right? Which can still change the pH of our bloodstream. But what happens here is that our muscle do have the ability to combine pyruvate with another waste product, which is ammonia, NH3, to form alanine, which is an amino acid. So my question is it safe to transport alanine in the bloodstream? Of course, yes, because all amino acids are natural buffers. Not only they are safe, but they can help resisting any pH change within the bloodstream. And therefore, alanine will be transported into the liver safely, and in the liver, it can be deaminated to produce pyruvate. And from the pyruvate, it can be used to produce the new glucose molecule by gluconeogenesis. So the liver is the only safe place by which we can safely get rid of ammonia, which is a toxic compound. So now you can see that we begin with two waste products, which is lactic acid, as well as the ammonia and the potential issue of acidosis, but we ended up removing those toxic compounds in the liver and having a pH stabilizer, which is alanine, that help to stabilize our blood pH. So this is such an intelligent design. And this process is known as the alanine cycle. For information, alanine cycle can only happen when there is oxygen to convert lactic acid into alanine, while Cori cycle will happen when there is no oxygen, which means our body is forced to transport lactic acid in the bloodstream, but we cannot do so in large quantities because of acidosis. And showing here, you have glycolysis, which is a process of breaking down glucose into pyruvate. So going down from glucose into pyruvate, you can see that there are three reactions. They are energetically favorable, which means they have very negative delta G. But when we try to reverse this process, these three steps are going to cause problems because from delta negative, now they become delta positive going upward. So my question is, can this reaction happen when they have a positive delta G values? Well, the answer is no. And for this reason, gluconeogenesis is not a simple reversal of glycolysis.